much. Thanks for your introduction. And I don't know, is this the best position or maybe I'll try to move around. So I think this is the best one. All right. This is basically what she said. This is who I am. And I studied the interface between these three. I'm more interested on fish and aquatic insects, and also studying their trends in this changing environment with climate change and things like that, with emerging stressors, and also more interested on the ecology part of them and things like that. So since you don't know me really, this is some of my previous work I've done. If you're interested, you could read that, but this is not what brought me here. So today I'm here to talk to you about the longitudinal changes of fish, assemblages, macroinvertebrates communities, and trace element contamination in this trace element distribution in this Afrotropical river called the Mara River. And some of the interesting fact about this river is that it's a transboundary river between Kenya and Tanzania. This is the river which feeds this ecosystem. So every day we wake up, this is what we wake up to when I'm doing my research somewhere in Kenya. So these are photos I've taken myself around this ecosystem just by driving around or when we wake up from our tents. So this is the amazing Masai Mara ecosystem, one of the major destinations for safari. If you guys are interested, talk to me. Well, you can be my research assistant over here. All right, so this river is really important because it's the one which it's like the lifeblood of all what is happening around this ecosystem. But by just looking at this photo, you can't really tell that there is this important river really which exists, right? And the thing is, a lot of studies really are focused on terrestrial ecosystem. Everybody talks more about the charismatic megafauna. But what about this, which is the most important ecosystem in this area? But a lot of people really are just seeing the water itself, the murky water, and nothing much. But what really lives in there, like what of interest am I really up looking for? So I'm shifting the narrative. If you saw my talk, it was from big to small. From big, everybody knows all what big animals are, and now we're shifting the talk more to the small cryptic and really endangered species in this important ecosystem. And some of the facts I've talked about in this river is why it's really the only perennial river system in this ecosystem, the Mara Serengeti ecosystem. And there's a lot of interesting fact about every year, annually we have the migration of these animals, which are called the wildebeest, and they're migrating from Tanzania side all the way to Kenyan side. And this is the river they cross. And while they are crossing, a lot of this is happening where a lot of them die within the system because probably they were too tired when crossing or they were attacked by crocodiles and things like that. Then what does this then do in terms of what I'm interested on the fish, on aquatic macroinvertebrate, on maybe aspect of trace element? Does this really influence all that in any way or not? It's a river also with lots of crocodiles, fauna, with lots of wildlife around us. As you can see, we are sampling with elephant on the background, with hippos behind us. And so, yeah, you should be now coming up with questions on how we do all that, right? How we survive. All right, so that brings us to what I'm really interested on my dissertation, and I've mentioned about this severally. It's more about just interested on this aspect of how these communities are changing of both aquatic biodiversity in this important river in the face of these different stressors, anthropogenic pollution, land use changes, and things like that. Where are we in terms of biodiversity for sure? And is all what is happening in the catchment affecting the trace element contamination in the river? Because if you saw a fish like this big fish, which people really are fishing there and they are eating this fish, does it have any contaminants in its body? And if it does, do those get to people really? So there's that aspect of human approach aspect where are we eating things which might be contaminated? Because the other last chapter of my dissertation looks at this contamination on these specific organisms. And so just I'm gonna bring you to speed what all this study all involves, and is like around globally, freshwater ecosystem are currently threatened by multiple stressors, and we have some literature on that. And these stressors they range from merely deforestation to agriculture to urbanization. Agriculture is more about what people are using during farming, the pesticides, herbicides, and all this. Because this catchment also, we have tea plantations, coffee, and people are using these elements as part of their farming, which if it rains easily, soil erosion, everything goes to the river. So that might find its way. The urbanization, as I mentioned, this river cuts across a very important ecosystem in these two countries where it's one of the major destinations for safari. So there are a lot of lodges, hotels along this catchment, and sometimes they tend to release effluent to this river because they don't really have well-equipped kind of waste treatment. So that somehow gets its way to the river at some point. And as we all know, insects are really threatened somehow, and there's a specific taxa or community of these insects which tends to be more threatened, which basically this is just a scientific term to mean a mayfly. These are stonefly, we have caddisflies, and we have our favorite dragonflies. So those are just scientific taxas, but that's generally mayflies, stoneflies, caddisflies, and dragonflies tends to be more in decline globally, and there are some graphs on how these insects are really changing. So this is happening already, but it's changing when we don't know what is happening already in tropics. So how is this decline also being affected in the tropics? Because majorly this has been more studied in this more of these phases in Europe, North America, Asia, and new tropics is basically more Latin America, Central America, and that kind of biosphere. But in the tropics, then what's happening in terms of all these communities? I'm gonna introduce this concept at this point is the river continuum concept. For those who are not ecologists, what this means is just like when we have enough river flowing from upstream or from the mouth, from headwaters to the mouth, the communities of whatever is available in terms of fish or insects might change based on where we are at every specific point of the river. So at headwaters, we tend to have more trees happening, so they have closed canopy, and they are hence they shed a lot of leaves into these rivers, releasing a lot of food for the insects, and that's basically what provides energy for the insects in headwaters. And there we tend not to have a lot of primary productivity. That's what we have this equation where productivity over respiration is less than 1. What that basically means is that there's no photosynthesis or there's no productivity happening since this closed canopy, sunlight cannot really get through. When the river flows a little bit downstream and to an open landscape, then the trees are less, they open up, and the light can now get in. And at that point, photosynthesis can really occur, where we have productivity of algae and things like that, is where productivity of R now is greater than 1. At this point, the communities are changing. And so basically, the communities which are found in headwaters where we have closed canopy tends to be more eating on the available food materials from the leaf litter and others. So they break down that food, making it available for other organisms which are living at this stretch of the river. And the ones which are here also break down the food as it's more like this fisher cycle. So basically, it's just to introduce the idea that within a river, communities of biodiversities might change based on where you are. And so this concept was introduced more in temperate region and here in the US, but what happens in the tropics where now we have issues to do with seasonality, dry and wet season? Do they change these communities really? What the aspect of closed canopy, where more in tropics, the rivers really don't tend to be having a lot of canopy, and that is not available, then what does that do in terms of species distribution in that area? So what about this concept in a river where I'm studying, where my river already has this stressor happening? We have issues of land use, we have issues of subsidies into the river from wildlife, that's just as I showed, the wildebeest happening, and also the issues of anthropogenic disturbance. What happens if you couple all those together? What happens to the aquatic biodiversity then? Is there a trend? How are the trends really happening there? So this is just a conceptual framework of what my stressors in this catchment are and what I expect of my community of fish and macroinvertebrates to really change. The idea that land use, if you increase land use by people cutting down trees and a lot of encroachment into these protected areas, then you are going to have a lot of sedimentation going to the river, since now we don't have a lot of trees all into the soil, and when it rains, we are probably going to have a lot of these sediment going to the river. And that sediment might, in a way, increase issues to do with dissolved oxygen and all that. And that might also influence the communities. So every single stressor, based on either it's a wildlife subsidy, land use, and pollution, from these point sources might, in a way, impact all those communities. So I'm trying to figure out how all those different stressors really impact these communities of insects. So as I showed you beautiful photos of this ecosystem in the Mara, and the question is, people are more interested on these big animals, but does the biodiversity in this river really matter for sure? And in that case, I'm going to introduce some of the stressors already I've talked about. But at the moment, what we have been faced with in this catchment of the Mara ecosystem is that we have had over years issues to do with land use change. And that has been documented in previous papers published by one of our professors in the department, that over time, there's just been this increased sedimentation in the river, because people are cutting. This is somewhere deep down in the forest, but there were people living there. So now, some 5 years ago, 10 years ago, people were living in this main catchment of this river, but the government intervened and they've been pushed away. And now the forest is trying to rehabilitate itself and going to its natural state. But that was like what was going on for the past 5-10 years ago. And this river throughout the year, this is the color of the water itself, even from the headwaters. That can tell you the impacts of what has really been going on in this catchment. We are having an issue with artisanal gold mining, and this is mostly in the downstream catchment of this river, somewhere in Tanzania, where people are mining gold, and they are using mercury as an element for amalgamation when you have to wash your gold. And that seeps easily to this main channel of the river. We have issues with dissolved organic carbon, where basically what that means is that increased carbon can be gotten from things like either terrestrial or dying animals, things which really add into more carbon. And one classical example was when these animals are migrating to the Kenyan side, a lot of them are dying in this ecosystem, and that might increase the dissolved carbon in the river. And there is some documentation that having dissolved carbon in the river can modulate the presence of mercury. If somewhere like in Tanzania, where we have artisanal gold happening, then how can the DOC itself modulate that available mercury itself in the river? So there are a lot of concepts, but I'll try to synthesize them and take you to one direction. Due to seasonality, we have this happening in this river every single year, that we have mass mortality of fishes at a certain period of the year. And that can be explained by things to do with more during dry season, the river is really low, and water it's only available in small pools of this river. So it's not like a continuous flowing river. And what is happening, since this river has like greater than 4,000 population of people, what they do, these animals, they're just grazing during the day, spending the rest of the year. They graze at night, they spend the rest of the day just in these pools, and that's where they are doing their defecation, urination, and that kind of chemistry really influences that pool. So that pool only has characteristics of elements which are really not good to the organisms themselves. And when we have flash floods happening in any certain period of the year, because those organisms were more used to this toxic product, and now it rains and washes down everything, we are having this kind of an event happening where we have a lot of these fishes really die. And some of it, I forgot to mention that Mara River itself has some of the critically endangered fish, like Labeo victorianus is the scientific name of the fish, but the common fish is the needlefish. What critically endangered fish, for those who don't know what that means, that basically means it's listed under the IUCN red list. when any species is listed in that red list, it really gives a signal that, wow, we need to conserve that species because it's dying and it's only endemic in this river. So if we lose all these kind of population in the river, then we end up losing that. Like, if you ever believe in dragons and things like that, if they ever existed, then that's how we can lose our fish. So also the issue of anthropogenic pollution, as I mentioned, there are a lot of catchments of tea plantations where people are doing that coffee, and they're using a lot of those herbicides and the pesticides to carry their own activity. And that somehow, because this is a slopy kind of topography of this river, that can easily bring stuff to the river itself. And so currently what I'm doing, I'm studying this river, both in Kenya and Tanzanian side, the different dots. The dots color shows you where I'm sampling. In Kenya, the 9 sites are all distributed from upstream of that river all the way to the border of Kenya and Tanzania, where is somewhere around here. We have a border somewhere around there, and all these other 3 sites are in Tanzania. So over time, I started my PhD back in 2021, January, after onset of COVID, and I've been sampling these sites every year. So right now I should be 3 times already. I've already done sampling in Kenyan side, but in Tanzanian side, I haven't been successful getting the permits, but this time round looks a little bit promising, and I've already applied, and we are already reviewing the applications. And the idea is to spend my spring 2024 trying to get samples from this side, because that's the aspect of seasonality. The only time I've been in Kenya sampling is usually during summertime, and that's just a particular one season in Kenya, and I've never gotten a chance to sample during this other season because I'm usually at UF, and because I have to teach around here, I cannot travel to go collect data, but this time round it's something which looks feasible for sure. And I'm just gonna introduce my 3 chapters and what I'm really doing in this study, and they are all grouped into 3. I'm having a chapter focusing more on fish, another chapter focusing on aquatic insects, the macroinvertebrates, and the last chapter focusing more on ecotoxicology of what is happening in terms of trace element contamination in this river, both in fish, macroinvertebrates, in the water column itself, and the sediment. So do we have any potential contaminants in the river, and where are we at in that particular point? So if you're fish lover, or you love insects, or you love ecotoxicology, I got you covered. So just sit back and enjoy. So for the fish, we are sampling fish using different techniques in this system, and one technique complements the other, because there are some species which you cannot get, you can't trap them or capture them using a particular technique. That's why you have to use these multiple techniques for you to maximize on your catch. And once we get this fish, what are we interested in? We are more interested in documenting the diversity in this catchment, as well as abundance, and also interested in the aspect of the gut contents, where the gut contents will give us more details on what these fishes are feeding, and is their food changing during this seasonality change, during dry and wet season. And you can use that for management purposes. Like, we need to ensure the water in this river does not drop to this level, because if it does, it might make this fish start feeding on this, which is not good. So there are all these different dynamics. But for today, I'm just gonna present a little bit of what we are finding in terms of diversity and abundance in this river. And interestingly, this is a site somewhere deep in the forest. This is our first site, headwaters somewhere in the forest. And there's no fish at that site, where the temperature drops to over there we work with degrees Celsius, my bad. But I don't know what a less than 10 degrees Celsius looks for you in terms of Fahrenheit. But in a river where we have less than 10 degrees Celsius, that tells you it's really cold in the tropics. That's a cold river for sure. So this is a site where the water is really cold and we don't have any fish there. We've done these numerous surveys, and the question is, why not? There are no weirs, there's no damming, there's nothing. We are not getting fish there. But that's where the aspect of sometimes when you're doing your research, talking to the local community can really be so informative. I just happened to ask one of the guys who come around, look for, they are really so interested to just onlookers when you have to do sampling. I was like, there's no fish here. What do you think? It was like, That's easy. And the guy was like, this site, he knows the site. Since he lives here, I was like, here, this site, which is the second side of the forest, that guy lives closer there because nobody lives in the forest. And he was like, if you sample it where I come from, that site will get fish. And these fish are moving upward, upstream, heading to site one where we don't have fish. But there's a point where their presence only reaches there. And the guy has not really gone to school, but he was like, I think because these guys fetch water there, they bath in this river, he was like, the water temperature there is cold. And these fishes are only up to the point where the water is warm. And above there, they don't like that, but they are starting to move up. I was like, all right, that makes a lot of sense. And actually, that explains why we don't have fish there. So the water really is really cold, and fishes are only occurring where at a particular point where they feel the temperature is okay for them, but they have started moving upwards. But to summarize this, is like we are having these two dominant species happening. There's labeo babas altinalis, is this fish up here. I'm catching this fish, but I don't want to move because we are recording. And labeo victorianus is the critically endangered fish, which is this guy here. So these are the guys who are critically. But in summary, these two communities of fish are like the most abundant in this catchment, and their population are fluctuating all over these sites. Gravias is common, is this fish here, which a lot of local community are fishing. There's a fish we caught, I think this one, which was close to 15 kilos, which is like really good weight for fish to be eaten by people who fish. And that's just the trend of all that. And but that's just basically to show that those two species really are like the key dominant in all those sites. The labeo victorianus, which is this critically endangered fish in all the sites, how is it doing? So there wasn't in site 1 and 3, there was no that fish, which is critically endangered, but the rest of the site, except 7 and these 3 sites, they didn't have the fish, but their abundance is really doing well. So it's somewhere around 50 divintos per site, which we caught. And the idea is you just catch the fish, you measure different matrices, and you release the fish back to the river, unless you are taking samples such as isotopes or things like trace muscles for trace element. So the fishes which we kill, you mainly using MS22 and all these well-structured protocol on how to kill fish. So if the fish, activists here don't come fighting me. We have really nice protocol on how to do it. We just don't throw it down and just cut it instead. No, they are really structured. But we take samples, muscles for trace element, and especially for gut, because there's no way you're gonna. There are more specialized new way using the new technology nowadays by tracers, isotopes, and things like that to learn what these fishes are eating. But that's more sophisticated kind of technique, and it might be expensive. So we taking some of these few individuals and just dissecting them and taking the gut out, and we are preparing slides to just learn what these fishes are eating because it's important. So the other aspect we wanted to see the diversity, and these are the diversity of these different fish changes across these 9 sites. But it's interestingly to note that this diversity starts going up between these sites, site 6 and 7. And from our map, something I forgot to mention, in this map, site 6 through 7 through 9 is basically where the migration of the wildebeest is happening. If that can ring a bell, what that means is that between this site and that site, these are sites. The wildebeest don't cross at just one particular point, but they utilize this space across whatever kilometers, and they are crossing those ponds. So what that infers to you, why is diversity increasing in this site? So that might infer, not yet, but that might indicate that in the sites where we are having the migration of wildebeest happening, probably we have increased food availability. For instance, when these animals are dying in the river, there are fishes which are good with eating the flesh of those, like catfish. The big fish I caught was like, it really needs that food. And it might be just a way that when these fishes move there, other communities try to colonize that kind of area when we have availability of everything, because their presence and their decomposition brings a different chemistry in the water. And that's where we are finding the higher diversity. So that might be one of the points which might be inferred by my results, but we are still trying to think on alternative. So I just put a graph of this diversity here with how the temperature, dissolved oxygen was changing. Why dissolved oxygen? It's really important for fishes. And a fish can only occur where the dissolved oxygen is about 5 milligram per liter. And that because basically it needs oxygen to breathe. So if there's not enough oxygen then. So this is how the trend is. But basically in this river, this is just a summary that we are having dissolved oxygen in levels that are within the limits, and fishes can really survive. Even if it is somewhere in Tanzania or wherever that fish gonna be, if this was to go all the way to this Mara River flows all the way to Lake Victoria, if you have heard Lake Victoria, which is one of the biggest freshwater lake. So if we extended this river somewhere all the way to the lake, is that whenever the fishes are occurring, it's good dissolved oxygen where they can all survive. That's good. But this can always drop based on the seasonality and things like that. I wanted just to see what is important or what makes these communities then appear there. For those who are more into social science, this is called a principal component analysis, and what it does, it combines all the different variables you measured at every site. Maybe it was elevation of the river at that particular point, maybe it's dissolved oxygen at every point. So it combines all that data and gives you this beautiful graph here to just show you that the sites which these dots here in fast sites, and that's why I had to put these two. So, but basically what is happening, where we are having these arrows longer, the elevation is like the main driver, which is like, makes sense, because if you have this graph, the first site somewhere, where is my first site somewhere deep in the forest, I imagine that the elevation of the altitude is really high and the river flows following elevation. So topography is really a great point. So what that means is that the sites here are having elevated levels of dissolved oxygen and elevation, and they have the opposite of all these variables happening here. For instance, site one, which is the main headwaters of this river, has high elevation, but also it has low depth, it has low phosphate levels, it has low turbidity, it has low river wing. That makes sense? But if you came to these sites, which are somewhere downstream, as you move downstream, the river gets really big, and that's where we are getting the river having bigger wings. But in that, so the river wing, which is bigger, is traded off by low elevation, because you don't imagine that site will be up there somewhere where elevation, so it's somewhere lowland. So basically, the distribution of these communities of the fish might be influenced by some of these variables happening here. And it's more like the sites which are downstream, where we have found higher diversity, are having higher turbidity, they are having higher phosphate, they are having higher depth, there is higher the DOC, just as I presumed with my wildebeest crossing. The sites which are downstream are having elevated levels of all that, and that might be the reason why we are probably having higher diversity happening downstream. So that was just my chapter 1 on fish. Sorry, guys, I'm doing a lot. So this second chapter goes much on invertebrates themselves and the insects. Why are they important? People have been using both fish and insects to monitor river ecosystem. Even in the US here in the temperate region, there's just specific organizations which deal with monitoring the river by not measuring the chemistry of the nutrients, but they only use these indicate organisms. They sample these organisms, and then they can infer if the river health is really good or not. So there's like a chart which gives you a green, a red, and all that, which when you find these communities here, that tells me the water is excellent for ecological purposes and things like that. So that's why they are divided into 2 chapters. These are some of the insects which I've taken with a microscope through my phone, and just to show you how they look like if you don't know what macroinvertebrates are. And we are sampling them using different techniques, using kick netting, and I'm following a protocol called SAS5. It's basically South African scoring system, where they recommend you sample these 3 different biotopes within a river, and biotopes is just habitats or where where a system doesn't look like the other. For instance, stones. If you go to a river, we might just find an habitat where we have lots of cobblestones, small stones. That's like an habitat in itself. Another different one will be GSM, which basically abbreviates for gravel, sand, and mud. Where you go to a river and you find that kind of a setup, that's a different biotop. And lastly, vegetation in themselves. If you've been to a river and just like disturb the vegetation, the riparian vegetation, or even aquatic vegetation, you might find it's having some unique insects or taxa compared to the others. So those are the main three biotopes: stones, GSM, and vegetation. And we are collecting all those sites. This is somewhere now deep in the forest where you cannot go alone, because continuously there have been issues with the local community. They were once living there, and now they've been told to move out, and they've been having pressure always to come back. So we have these guys who are always guarding this forest and keeping it safe. So you can never go to the deeper forest without. They are called the KFS, which abbreviates Kenya Forest Rangers and Service. And it's like you have to have these armed guys to go in there to sample, because anything bad can happen anytime. So just to put that into context. But yeah, so we are collecting them, sorting them into their different groups, and I do the whole job of identifying them to their different groups, because I'm trained on this. And also, interestingly, you can see this nice graph just like of the fish. Is also like the macroinvertebrates are also more responding towards these sites where we have in our wildebeest crossing. It's where we are getting more abundance, and even like the diversity is changing towards the downstream sites, which it's really like not the usual trend in aquatic systems. So if you inverted this and you had site 1, 2, 3, and you showed me this, I'll be like, this is really good results. Because but at this point, when it looked like that, is like having a lot of questions. I have to go back to the field and try to figure out why is it so. Because the sites which are downstream, as you saw from the earlier PCA chart, they have higher turbidity, they have higher nutrients, they have higher everything. But still, the other sites which harbor a lot of this biodiversity, and it should be inverse true, that the sites with less of all those elements are the ones which should have a lot of biodiversity. So it's really interesting system. And that's why it's sending me again to the field and trying to like figure out it's not obeying the common law of ecology. This is going opposite of that. And with this, I'm interested to compare. So Amanda, who is happened to be my advisor, she's been working in Kenya for the past 10 years. I met her sometimes. That's a story for a different day. But they've been collecting data in this river for the past 10 years. So I have that data already. And I'm currently also collecting macroinvertebrates. And the question is, with these two periods of this data collection, compare in terms of what species of macroinvertebrates we are finding in the river. This is a PCO, which means principal coordinate analysis. It's good with giving you this visualization to see things in the moment. This is my historical data here. My historical data, which was collected 10 years in these 4 different sites. This is my current sampling of the same sites presently. So this is 10 years, but what that is showing you that 10 years ago, the sites, so that it measures the distance between these sites. So these sites are much closer to each other, which means these communities and species were not so different from each other in all these sites. And that's the time when, probably 10 years, we did not have people living in the forest. They started living there like 5 years ago, and they were sent out like previously. So this captures the effect with no humans in the forest, if that makes sense. And currently, I've sampled 10 years where we have had that impact of people going to the main source of this river. What is happening? We are seeing our sites are moving farther apart. What does that mean? That means that the communities or even the species have changed within the sites and also from what was sampled. So if my site composition was the same as past composition, these names will be closer to each other. But also the fact that they are away from each other, that shows you things have changed. That they are totally different from what it used to be there 10 years ago and what is there. And the issue is what has made this change? What is happening? That has really changed the composition of all that. So I went further and wanted to compare between the sites. So remember what I showed you within a river: upstream, middle reach, downstream. As you move in these different sections of the river, communities are expected to change in a certain way. So is that the case with this? Because this is basically what it shows, that within these different reaches of the river, the summary here is that species are not really so different within these reaches. If you are in upstream or middle reach or downstream, especially these two, the upstream and middle one tends to be more together. Their species are different, not that they are similar to each other at least, which makes a lot of sense since those are the most sensitive taxa within the upstream catchment of the river, and the downstream tends to have more intolerant or not really sensitive taxa, which might be any other taxa which is tolerant to pollution, which this bigger circle of red shows you that, oh, well, in downstream, it's a little bit different. We are different from these guys who are in here. So that shows that. The seasonality part of it, if you sample during dry season and wet season, that does it really have any change and differences? This summary shows that there's no actually clear differences between if we are in wet or dry season. So seasonality part of it, probably in tropics, doesn't play really a big role. I did not go in this river to come up with this or discover what is happening. So what you are seeing right now, this is an organism. There are two of them. This is this guy. This guy is a mayfly. His name is trichorhyd. And this guy is a diptera or a fly, and his name is simulidae. These are pupil, these are larval stage. What is happening here, this is something called phoretic or phorecy association. For those who are not familiar with this, you could Google and learn more about what that term means. But basically, this organism cannot live on its own in this river. So it has to find a mechanism to depend on the other guy for its survival, right? So a lot of these simulids are attaching themselves. So this guy is really a bennett on someone's head. And it's like, I have to live there for some time. You have just to allow me to be with you for some time. And all these organisms, this is their larval state. But when they grow adults, they develop wings and fly out of this river. And they are like the flies you see when you go for camping. When you go for camping and you light your lights at night at camp, those are the kind of flies which are attracted. So it's a nice association of a nice discovery in this river. And we are trying to write a short communication of all these evidence of this is happening because it's not well documented, especially in tropics. And we have really positive information about this. But this is really something cool we found in this river as well. That chapter, which is my last one, I don't want to lose you, but this is where now you belong because this is the aspect of humans, social science with humans, that right? So my fish, my insects, how do they relate with humans then? So this, I'm trying to measure these levels of toxicants in the bodies of these organisms, both fish and all that. And more interest is the fish. And I'm collecting all these different samples. Both I'm wanting to measure what are the levels on the water column, what are the levels on the sediment, because the sediment can act as a source or sink based on these different aspects. And the sediment itself harbors invertebrates, which are also trying to scrub and feed on that sediment. And also that those insects are being fed on by fish. And people tend to love fish, and they're also eating the fish. So the question is, any of these really is contaminated based on trace elements? So trace elements are basically these elements which are not nutrients. So I'm interested on something like lead, cadmium, arsenic, mercury, because we've had in Tanzania there's mercury, and all these magnesium, name them, sodium, and all those different elements. So something interesting from the river is that all these, how we take these into the different fish and take the muscle tissue and all that. But we do a pre-treatment of these samples. We dry them in Kenya before I fly them to the US. We are analyzing our sample at Center for Environment and Human Toxicity. It's somewhere along Garalame way there. And something interesting, this is water, sediment, that's gonna be on fish and macroinvertebrates. So paying attention to what levels there exist in water, all right? So the water column tends not to have a lot of elements, and that makes a lot of sense. So these elements can either be coming from geology of this area, that's one aspect, or it can be more on point sources of pollution. So the many launches along this river, or people just carelessly trying to dump stuff to the river, that's what might bring these levels in the river, but they cannot be naturally occurring in the water itself. The water should be free of these elements, but so it cannot just be naturally occurring. And the thing is, in water, we're only having higher elevated level of this metal. For those who missed their periodic class, chemistry class somewhere in university or high school, ST, this element SR, that's strontium, for those who missed the class. But strontium is typically elevated level just because look where that is happening. So the sites where we have in the wildebeest crossing is where we have in strontium. Strontium is proximal like carbon. So when these guys are decomposing and all that, they are releasing these elements in their bodies, these wildebeest. So the strontium, it's more elevated in downstream side. In the aspect of the sediment, we are having the same metals, but now lead is actually leading here. So we are having now presence of lead happening. So these units are in micrograms per gram. Microgram per gram, which is substantial, good amounts in sediment. So sediment will have high elevated levels since it's just storing everything. Everything, when it comes to the catchment, it sits on the sediment. So the sediment really is like the main source of all that pollution. But look, with our fish and macroinvertebrate, the same element, same scale. So it's surprisingly that the insects themselves are having higher levels of this stress element as compared to the fish. Who might have a guess as to why that is so? So insects have different varied feeding modes. There are some which are shredders, others are scrappers, others are feeding on the others, and others are more like collector-gatherers. Those are like generally the four classification of insects. So if you are a scrapper, what you are doing, you are seated there in the river and scrapping things off the sediment, which means sediment add higher levels of those elements, and the one in contact immediately feeding on that. So the insects tends to accumulate more of those elements compared to the fish, which gonna come chasing me afterwards after I've eaten my element. The fish will come chasing me and it will feed on me. So the fish has the potential to bioaccumulate, which means if today I eat 10 micrograms of your elements, tomorrow I eat again 10. So over the years, I'm probably gonna have high elevated levels. So it's gonna be the accumulation of all those 10 over time. So this, in summary, what this shows you, our insects have higher elements. These are parts per billion, which really in trace element or even in concentration, parts per billion is really, really small. But if I change this scale, since there are a lot of zeros, I could upscale that scale to something like microgram per gram by dividing it by 1,000. That still shows substantial levels of those elements in insects. And the insects are being fed by the fish. So the take-home message is, at the moment, the levels are still within the permissible levels. But what will happen over time with issues to do with climate change or seasonality when this river doesn't have really adequate water in this dry season, and what will happen to those elements then? And people are still feeding on this fish. So at some point, people might end up feeding on fishes which have higher contamination levels based on these different stressors, the seasonality aspect and all this. And with that, I'm intending to do the impact of my study where I want to conduct workshop sessions in Kenya. I've already identified 3 universities which are going to be working, and the choice of the universities was based on this catchment of the Mara River. The ones who really directly benefit from this catchment are the ones whom they can use this catchment for their studies. So I have 3 universities where I'll be going to teach all go and train students there, especially the undergrads, more about how insects are really important. So we're going to be having it in different sessions. We're going to be having lectures. We're going to have a field trip where we're going to go do our stick netting and bring the insect where we're going to identify them with microscope. And the idea is to encourage these students and more to appreciate what we have in these rivers, and also take up more in future for their graduate school, some courses which might also have an impact on this ecosystem. So that I'm happy to mention that this actually has been funded by TCD. I recently got an email about the practitioner grant, which I'd applied to, and I'll be going to Kenya in spring, and that's one of the things I'll be doing over there during that time. Still, same time, I'm trying to get to Tanzania and get these sites where I haven't measured all these effects, where we have mostly these levels of mercury being more potentially happening there. So that might be potentially be happening during that time. And with that, I want to thank all these different funding organizations, the permitting process and all that, and all those amazing folks who have made this work come to reality. And welcome.